I conceived of the Window Therapeutics platform over 10 years ago, and in the time since, I've been developing it in my laboratory in the Department of Chemistry at MIT and through my affiliation with the Koch Institute for Integrative Cancer Research, where we've been developing these brush prodrugs for a variety of different cancers. And this work has received over $12 million in funding from a variety of sources, National Institutes of Health, DARPA, the National Science Foundation, and many others, including industry partnerships.
At Window Therapeutics, our modular design is only possible because of decades of Nobel Prize winning research that has led to chemical structures that are impossible until very, very recently.
I mean, it's different in almost every way, particularly at the molecular level and the way we approach the design and the synthesis, the manufacturing of these molecules. So to say 10 years ago, the Window Therapeutics prodrug platform could not have been made. The synthetic methods did not exist in the field of chemistry to do that. But through leveraging the latest technologies, we were able to now design this prodrug platform that has this bottle brush molecular shape, which comprises a polymer backbone that has at every repeating unit a covalently attached drug molecule, where the linker that attaches that drug molecule, we designed using our expertise in synthetic chemistry to cleave and release a drug only in response to cues that are given by cancer cells or in the cancer microenvironment. What's very important is that these drug molecules that are attached to the polymer backbone are shielded within a shell of polyethylene glycol, which is a biocompatible polymer that's used in a number of different medicines today. And it's this shielding that protects the drugs on the backbone from premature exposure and release in healthy tissues. Our molecules have sizes in the range of 5 to 15 nanometers or so, which is on the same size scale as many of the biomolecules in your body right now. Proteins, for example, mRNA molecules, things of that nature. So we have this unique ability to make these prodrugs at a size range that nature already optimized to achieve optimal tissue and cell targeting. So with the antibody serving as an active targeting agent to be able to bind to a given cell, this can bring our brush prodrug to that diseased tissue and allow us to further improve the therapeutic window. Two additional very unique factors of our platform are one, that because of our manufacturing approach and the modularity of the synthesis of these molecules, we can take not just one drug, but we can take 2 drugs, 3 drugs, or really any arbitrary number of drugs and incorporate them into our platform in a precise ratio so that we can deliver combinations of drugs to individual cancer cells or tumors with precision and control, which really no other system can do today. And this allows us to take advantage of synergistic pathways in cells where maybe a combination of 3 drugs is more effective toward a cancer cell than even the sum of each of those drugs together. And last but not least, the way we approach the manufacturing of our platform allows us to readily conjugate our brush polymers onto biomolecules such as antibodies, which allows us a completely new way to approach the targeted delivery of our compounds.
I think the brush technology has the ability to not only utilize conventional agents, targeted and immune agents alone or in combination, but to have delivery to the patient at the site of their particular cancer with the appropriate distribution of medicine so that cancer cells are targeted and normal cells are spared.
At the moment, combination therapy faces a long-standing translation problem from the benchtop to the bedside. And this is because these therapies use a multitude of different component drugs. Using the Window platform, we can now incorporate all of these different drugs onto the same single carrier system, allowing us to A, distribute uniformly into the diseased tissues at the same time, and B, program these release kinetics. With Window, all of this other drug-like properties is offloaded onto our platform, leaving the drugs, only one task is to eradicate cancer cells. That said, we can now simplify the entire process of drug discovery potentially and allow us to access not only drugs that is already approved, drug that's already currently going into clinical trials, but also drug that is most much earlier in the discovery process. The optimal drugs for window might have already been discovered and discarded because of non-optimal other properties.
We've taken a bottom-up approach where we've modified and advanced our chemical platform so that it can be applicable to all forms of drugs. What we are able to do is we can come in at every single stage of drug development, from the drug discovery phase to the preclinical phase to the clinical phase, to make it so that drugs that are normally suffering due to poor ADME properties or excessive toxicity can be revitalized by using our special window therapeutics platform.
Particularly with the onset of novel immune therapies, we're targeting and selectively activating the right mechanisms within the greatest disease-fighting machine ever invented. The human immune system has tremendous potential. When we think about the same potential for other diseases that have been continually frustrated and impeded by these same fundamental characteristics of pharmaceutical properties, there's even greater promise.